Ever wonder why we have different blood types? Is it just a random quirk or does it serve a purpose? For many years, blood type identification wasn't possible. It wasn't until the 1900s that a young researcher in Vienna named Karl Landsteiner proposed the idea that some blood reacted poorly to others due to inherent differences in the blood makeup. Landsteiner's tests led to the confirmation of blood types A, B, and O, with type AB being shortly discovered after. For his groundbreaking work, he later was awarded the Nobel Prize. So, what makes these blood types so different? Blood types are determined by specific antigens on the surface of the red blood cells. Antigens are proteins that trigger immune responses. The ABO system is the most important blood group system in human blood transfusion. It's determined by the presence or absence of antigens A and B on the surface of the red blood cells. Type A. Type A has A antigens on the surface of the red blood cells and anti-B antibodies in the plasma. Type B. Type B has B antigens on the surface of the red blood cells and anti-A antibodies in the plasma. Type AB has both A and B antigens on the surface of the red blood cells and no anti-A or anti-B antibodies in the plasma. Type O has no antigens on the surface of the red blood cells but has both anti-A and anti-B antibodies in the plasma. The RH system is another critical blood group system named because the early experiments describing it were done with rhesus monkey blood. It is also referred to as the D antigen. RH positive. If your blood has the D antigen, then you are RH positive. RH negative. If your blood lacks the D antigen, you are RH negative. Evolution. Blood types evolved as a defense mechanism against diseases and infections. Researchers have noted that type O seems to have evolved before humans migrated from Africa and is particularly prevalent in areas known as malaria endemic regions. Knowing what we do about numerous changes in red blood cells that evolved as a means to avoid this deadly disease, scientists are considering the real possibility that malaria and potentially other diseases gave early human blood groups the push they needed to evolve. Malaria, particularly caused by Plasmodium falciparum, has shaped the prevalence of blood types. In people carrying the parasite that causes malaria, infected red blood cells pile up in small blood vessels, blocking blood and the oxygen it carries from getting to the brain. But people with Group O blood type have significant protection against malaria. A 2007 study in the journal Proceedings of the National Academy of Sciences, for example, found that people with type O blood were 66% less likely to develop severe anemia than people with other blood types. The A and B antigens both evolved over 20 million years ago. Their exact purpose is unknown, but they may play a role in blood clotting and help to protect against certain diseases such as cholera. Besides the main blood group antigens, there are 15 other types, such as the Duffy group, which offers resistance to certain malaria parasites. People who lack the Duffy antigen are relatively resistant to one of the two major malaria parasites, but it is rarely seen elsewhere in the world. The distribution of blood types also correlates with susceptibility to other diseases. For example, people with type O blood are more likely to have cholera, plague, tuberculosis, and mumps, while those with type AB blood are more prone to smallpox and salmonella infections. So, blood types have evolved primarily as a survival mechanism against diseases. That is why we have different blood types. It's all about survival and immunity.